This morning I attended the clonal hematopoiesis session, and I'm generally very excited about this field. I feel that this is a way to prevent leukemia, if at least in subset of patients where we can actually discover this preleukemic lesions. So there were several lectures there, and so the first one was from Sid Jaiswal from Stanford. And his paper just came out in Nature. He identified polymorphism in the gene called TCL1A. So people who have one specific type of polymorphism, this accelerates the mutant clones. So they show that people who don't have this particular polymorphism, the clone size stays kind of stable, but if you do, then the clones expand faster, and they propose tentative mechanism how that happens. So I think that's biologically very interesting observation. And then but not all the clones, so it was like mutation specific. And then the second speaker, Jennifer Trowbridge, she talked about specific subsets of CAH called DNMT3A mutant CAH, and she identified that this clonal cells secrete some cytokines that affect the microenvironment. And the microenvironment becomes senescence. And so senescence is something that I'm very interested in. I think it plays a big role also in the oncology, but they looked at senescence in microenvironment. And the end, which caught particular my attention, the senescence has been long associated with overexpression of end-apoptotic proteins such as BCL-XL. So they actually used the inhibitor navitoclax for BCL-XL, and they showed that they can suppress the senescence phenotype and suppress the expansion of the clone, although they did not show yet the, let's say, reduction of transformation or reduction of survival, like extension of survival. So these studies are still ongoing, but I think potentially it's very interesting because of course treating the patients who have clonal hematopoiesis is very difficult task because they are healthy individuals who have clones. So some of them may or may not transform towards acute MDS and AML, but all of us are like looking at the strategies. What are the safe drugs that can be used? So I think the use of navitoclax is very interesting, with the caveat that DNMT3A mutant CH has like a lowest propensity to transformation into AML, but still biologically is very interesting. And then the last talk was by Ben Ebert, who talked about the association of CH with the liver chronic conditions, and also about their recent work, which is about to be published, where they identified the clinical predictors of CH progression to AML. And so they identified the 10% of CH patients have 50% risk of progression to AML in the next 5 years. So that's like really like high, risk of progression. So I think these are the people we have to like focus on and treat them perhaps with some of the safe agents which we're trying to identify. So I think that was like the session that I enjoyed a lot.